Thank you very much, Jean-Claude Carrel, to have invited me, and also thanks to the scientific committee to have invited me here. Despite the fact I'm not a pediatrician, but an adult endocrinologist. So this slide shows the trend in obesity for adults worldwide. And so you see the tremendous increase in the number of obese patients everywhere. And probably there's about exactly the same trends in children. Now the question, everyone asks the same question, why? And maybe it's not the good question. The good question may be why we are not all obese, despite the fact that we share the same deleterious environment. And my own response, you can guess, is in genes. And that is an historical picture of identical twins and fraternal twins. And you can see that identical twins have about the same BMI. There are different evidence for a role of the gene. And I would like you to see this slide. This slide is a little bit complex. It showed the risk to be obese at the age of 25 when you are obese young. So if you see on the top age 1 and 2, if the child is overweight at 1 and 2, the risk is not enormous. But what is enormous for this child to be obese later, if parents are obese, as you can see here. And when you see and you follow children who are older at the age of 5, 6, 9, 10, or later, you see that when they are teens, the major risk is for children who are overweight, and the role of parents' obesity is less important. So the lesson here is that for young children, it's quite important when parents are obese to see if the child develops obesity or a little bit later in age, and what we can do. And we see later that we may have some responses. I've been asked to present the genetics of obesity, but same time I would like to discuss with you how useful is genomic medicine for obese children. In type 2 diabetes, or in fact what we call atypical diabetes, it has been shown for a very long time now that detecting this form of monogenic diabetes is quite important to improve the treatment of these children and adolescents. And the question is it the case for obesity? First question is how much genetic obesity is. So back to twins, there is a study that I think was quite important from UK, from Jane Wardle. Jane analyzed 5,000 twin pairs, aged 8 to 11, and she found a very important heritability of BMI and waist, about 70–75%. I would like you read the conclusion of a paper which I think is quite important, saying that, of course, the epidemic obesity is due to the change of our environment, but the individual differences between children can be attributed to genetic difference between these children. Now, the other question is, if obesity in children is genetics, what is the component of obesity that is more genetic? Is it energy expenditure? Is it appetite? And the conclusion from Jane and from other people are exactly the same. What is very important is appetite and food intake behavior. And there is different questionnaire, as you know, but depending on the questionnaire, you have an heritability between 60% and 80%. If you look for physical activity, you don't see something very significant for genetic factors. Does it mean that physical activity is not important? Not at all. There is one interesting study which was not in children but in adults, And this study, again, it's in Finland, in twins, analyzing heritability of fat mass, which is even more precise than BMI. And what they did is to have a precise evaluation about the physical activity of the twins. And they divided in different groups, lower physical activity, medium, and height. So what does it mean that for the twins, for the individuals who have a very low physical activity, their heritability is enormous, nearly 90%. Now, if you see the twins who have a high physical activity, you have a dramatic drop in the heritability of fat mass. You have different ways to interpret this result, the yin and the yang, but one way is that physical activity is able to modify the action of the gene for the predisposition of obesity, which means that even if you have everything to be obese, physical activity can change the situation if done early in life. Now, we know that obesity, like diabetes and many other diseases, is highly heterogeneous, genetically and clinically, of course. And you have now about the vast majority who have a polygenic form of obesity. We discuss that later. And we have the monogenic or monofactorial form of obesity. And as you know, there's the syndromic and the non-syndromic form of obesity. I don't have time, and I'm not expert in the syndromic form of obesity, but you all know that there's a lot of syndrome associated with obesity, like Bardet-Biedl syndrome, Cohen syndrome, and of course Prader-Willi and Prader-Willi-like, and Wegener. For me, the important thing to say as a geneticist is what is important when you have a child with obesity is not to see obesity, but to see the evidence for other phenotypes that can be related to a syndrome. Knowing that the definition of a syndrome is not always easy, I will show you later, but if you have an obese child and you have any evidence for one of these syndromes, it should be important then to look for molecular diagnosis. Now, the important other part of monogenic obesity are what we call the pure and non-syndromic form of monogenic obesity. The important information is people like me who work on different cohorts found about 3%, 2%, 5% of the cases had known mutation in one or mutation in one of the known genes for obesity. That's the first information. It's not extremely frequent, but that exists and shouldn't be neglected. The second important thing is that the phenotype is excessive hunger, hyperphagia, and they are all due to a dysfunction of appetite in society. An interesting thing is that when in the 90s, I began some work on that, nobody really believed something which is a little bit annoying, that human and rodent have exactly the same gene for obesity. And this slide showed that the same pathway, which is the leptin pathway, when impaired, leads to rodent obesity and human obesity, exactly the same gene. And I'm sorry, so probably most of you know that, but leptin is produced by the adipose tissue and circulate in the blood to the hypothalamus, arcuate nucleus, and bind to leptin receptor, and different things happen. And at the end of the day, there is a stimulation of anorexigenic effectors. And when something is wrong in this pathway, appetite increases, and there is obesity. This slide presents the same thing a little bit more precisely and showed the gene, the 10 genes that has been found so far to lead to monogenic obesity in this pathway. So you have the leptin, so produced by the fat, leptin receptor in the hypothalamus, POMC gene in the hypothalamus, proconvertase 1 and 3, also named PCSK1. And you have MC4 receptor, MRAP2, SIM1, BDNF, TRAC-B, and possibly and SH2B1. And all these genes or each of these genes, when mutated, can lead to about the same thing, extreme obesity associated with hyperphagia. But we will see that it's not so simple. So human obesity genes are the same than in mice, and all the genes, with no exception so far, there may be in the near future, but so far, are involved in the leptin-melanocortin pathway. Let's see some examples. So the most prevalent one by far is the melanocortin-4 receptor, MC4R. I told you that in different cohorts, maybe 2, maybe 4% of the patients, especially in children, have mutation in MC4 receptor. What you can see here in blue is a little bit surprising because it's said in a paper, in a review paper I published several years ago, I put autosomal dominant or recessive. Is it dominant? Is it recessive? And in fact, it depends on the population. I had the opportunity to work in a large cohort of several hundred patients from Pakistan, province of Lahore. All these patients are originally from consanguineous families, and we found a mutation, recessive mutation, so the 2 alleles are mutated in MC4 receptor. Which is amazing in this population is that the parents are not obese. Only the child and the parents are by definition heterozygote for MC4R mutation. So in Pakistan, you need 2 mutated allele, so it's recessive, to develop obesity, which is not the case in Europe. In Europe, if you have a child with a mutation, the penetrance, so the proportion of children who develop obesity is about 80%. So in Pakistan, you need 2 mutations to develop. In Europe, that is in France, but I think the same elsewhere. If you have only 1 mutation, you have 80% risk to develop obesity. But that doesn't mean that environment is not important. So we have published several years ago a paper showing that the penetrance, so we have analyzed 3-generation families with a mutation MC4 receptor, and we found that in children the penetrance was 80%, but in parents and grandparents it's much, much less. But they have exactly the same mutation. So we asked the parents and the grandparents their weight at the age of 20, and as you can see here, it's amazing because at the age of 20, only 10% of the parents and grandparents were overweight. So today, their grandson and daughter, they have 80% risk to be obese when they are children, but 1 or 2 generations ago, the risk was much, much lower. And in my view, that means that the environment has changed in 2 generations, and probably today to have this mutation is associated with a higher risk, probably not to the mutation, but probably due to the environment that help the mutation to express and help the development of obesity. Now, does it change something to detect this mutation? That is a study we just published in diabetes care. It's not about children, it's about adults for obvious reasons. And it's a bariatric operation. And we sequence the melanocortin-4 receptor. We analyze carefully feeding behavior and binge eating, for instance. And we found that knowing the presence of a mutation MC4 receptors and knowing abnormalities in behavior is quite important to predict the success of bariatric surgery, and especially the outcome in complication of the surgery. And the first and easy outcome is to predict what kind of, or to guess what kind of surgery you have to do, bypass or bending or sleeves. It's clearly bending, for instance, doesn't work in this kind of patients. So knowing this mutation can be useful for bariatric surgery. Now, the second example is very well known because it was the first gene found, were leptin and leptin receptor. So they are recessive. You need 2 mutations to have obesity. But what you see here in this review is it's not exactly the same. So the phenotypes are different. What we did at that time was only to summarize what was published by us or by others. But now we have some evidence that probably is not the case. And because these studies have been done in a very small number of subjects, have been compared to other obese people. In fact, when you take the opportunity of working in Pakistan, you have 2 important information. First of all, you realize that in this population with high level of consanguinity, the proportion of patients with monogenic obesity is enormous. So that's a paper we published in Obesity last year, and we found that at least 30% of these patients are explained by the known gene for obesity. So that's, for instance, that we found that 20% of these patients have mutations in the leptin gene, and 6.5% the leptin receptor, and all recessive. And as you can see here, if you compare, you have here the mutation, but if you compare the phenotype between these people and this, you have exactly the same phenotype. So personally, I believe that if you analyze exactly the same population in the same environment, you'll realize, which is maybe not a big surprise, that leptin mutation and leptin receptor mutation lead to exactly the same kind of phenotype. Now, is it important? I think it's important to know that because leptin is a cure that has been shown a long time ago by Steve O'Reilly and Sadaf Farooqi. Now, the drug is in the market, not for this purpose, but can be used. The sadness is this drug has been sold by one drug company to another company, et cetera, and I totally failed in the last 5 years to ask the new owners of the drug to produce the drug for the people in Pakistan. And so, because of course they won't make any money, so I was not able to cure the patient I had in Pakistan with recombinant leptin so far. Another important thing is the gene PCSK1. So it's peptidase proconvertase 1, which is quite important because POMC is a gene that produces a prepeptide, which is quite long, and then it's cut in different pieces, and one of the pieces is melanocortin, alpha and beta melanocortin. So if you have a deficiency in this enzyme activity, then you develop obesity. Now, we and others have worked for a very long time, and it's interesting to see the phenotype. So again, on this review, I put different things which seems to be incoherent. But why it's so incoherent and heterogeneous? And the reason is because it depends of the kind of mutation and the number of mutation. So 2 important things for PCSK1. If the worst phenotype is when you have 2 mutations, and this mutation completely kill the activity of the enzyme. That had been published originally by the group from Cambridge, and it's a terrible disease. And then, if you see here on the right and here, you see that if you have a less deleterious variant, you have some kind of malabsorptive diarrhea, reactive hypoglycemia, and severe and early onset obesity. When it's heterozygote, so only one highly deleterious variant, you still have a high risk for familial obesity. And when you have a SNP, a coding SNP, that has been formed by GWAS, then you have a mild increased risk for obesity and a very modest variation in fasting proinsulin and glucose. So the conclusion here is that the number of the nature of the mutated allele explain the severity of the phenotype and the association with other phenotype. So that's the conclusion. Conclusion is that the functional impact of mutation dictates the phenotype caused by this mutation, and the large variety of mutation you can find in the same gene are causing very different forms of obesity. So each case is different. We have shown exactly the same thing for an important gene named SIM1. It's a transcription factor. It's necessary for the development of the hypothalamus. And also, the phenotype are different. So originally, Amélie Bonnefond in my group worked on patients with Prader-Willi-like features. And she sequenced and found several mutations, as you can see here. The second thing we did in collaboration with Sadaf Faruqi and an Australian group was to analyze the effect of this mutation. That was not easy, but we eventually succeeded, and we found that only 3 mutations has a functional effect. So we did again the calculation, and we found that only the mutation with a functional effect were truly associated with obesity with an odds ratio of nearly 30. So it's quite important. For the others that were not associated with obesity. Now, you have exactly the same information I gave you for PCSK1. The nature of the mutation tells you the risk for obesity, which means that finding a mutation in SIM1 is not sufficient to tell you if the mutation is really responsible for obesity. But it's even more complex. We have a large family for these 3 mutations, and as you can see here, you have, with exactly the same mutation, some people are only slightly overweight, some have a morbid obesity and nothing else, and you have some children with Prader-Willi-like, and you know what does it mean? So that means that they have, in addition of obesity, many other phenotypes that probably due to an effect, a deleterious effect in the brain function. POMC is quite an important thing. It was one of the first genes that has been found by our chairman today. A long time ago, I was fascinated by the picture of the red hair pigmentation of the first patient she published in Nature, if I remember, or Nature Genetics, one of these important journals. And it's quite important because POMC produces MSH. MSH binds MC4 receptor. If you have a mutation in POMC, you cannot produce MSH. But the idea was to find a drug that will directly stimulate the MC4 receptor signaling, bypassing POMC. And that's exactly that our chairman did recently when she published a month ago or 2 months ago this paper in the New England Journal of Medicine showing that an agonist MC4 receptor named setmelatonide, difficulty to remember the name, but there are 2 patients who were extremely obese, as you can see here. And you see the dramatic effect of this drug on the weight. That exactly reminds me of the paper on the leptin treatment for the patient from Cambridge who had received this drug. So it's a very dramatic effect. I don't know exactly if there's some side effect, it will be for long, but it's the first time to my knowledge that a drug has been, which is not natural leptin, that has been produced and can have such a dramatic effect on. And if you read the last paragraph of this paper, our chairman wrote that it might be useful for other monogenic defects. She mentioned leptin receptor, PCSK1, and she also mentioned Prader-Willi syndrome. And I don't know what would be, if it's true, or if there are some evidence already, or it's only a speculation. But if it's the case, it's really a revolution in the treatment of obesity. So the environment is modifying the penetrant expression of the gene for monogenic obesity. In consanguineous population, a large proportion of patients have non-mutation, and we are doing now exome sequencing and genome sequencing, and we have additional genes, we are working on it, that may increase the number of genes and the understanding of the physiology, which means that if you ask the father and mother if they are first cousin, you may have a clue to detect this kind of abnormality and a reason for sequencing. And there are now some treatment that can be useful to cure this patient, I don't know, but at least to really improve this patient with this kind of homozygote mutations. Now, there's not only point mutation. A large part of the genetic polymorphism between individuals are copy number variant. A different kind of copy number variant, but at least if I want to be schematic, I will say that there are some deletion, less DNA, duplication, too much DNA, and you have some gene which exists in a form where there has 1, 2, 3, 4, 5 different copies of the same gene. What has been shown in the recent part is that at least some large copy number variants are present in few individuals, but confer a high risk for obesity. The best example is half a megabase copy number variant on chromosome 16p. 1 every 2,000 individuals have a deletion, but 1 every 2,000 as well have a duplication, so more than two copies of this region. In 2000, and I don't remember, 6 or 7, I found 2 patients with that. One in Northwood Park in London, one in the Catholic University of Lille, and we say, oh, maybe it's due to obesity. Then I contacted my friend from Switzerland, Jackie Beckman, and Sébastien Jacquemont, and Alexander Raymond, and they had exactly the same result. And then we analyzed by the GWAS, so it was 2009, sorry. We analyzed the GWAS data we have, and that's what we found. We found that that are children, and children with this deletion on chromosome 16, half of them are obese, half of them are normal weight. But after the age of 20, nearly everyone is obese and extremely obese. So there is an enormous risk to develop morbid obesity in adulthood when you have this deletion. So it's quite important, even if the child is not obese yet, to try to detect that, especially because there are some other problems. They have autism or other psychological problems, with also, it seems, a decrease in the IQ and learning disability for many of these children. Why it's important? That's some results we have. And basically, I will say that the gastric banding doesn't work in this subject, and even the bypass doesn't work all the time, and we have to be extremely careful before proposing this kind of bariatric surgery to this kind of patients. Now, I told you that when you have a deletion, the risk is enormous to develop obesity. What about exactly the opposite? And we published a year later, in 2011, another paper when we showed that the patient with a duplication, so more DNA, are underweight. And that's some data we had originally, and you can see that nobody is obese with this duplication. But please see for the young age, and you see that there's a lot of children with this duplication who are really skinny, and for some of them, what was said on their file that they had a failure to thrive. And we found about 5–10% of the subject we analyzed with a failure to thrive having this duplication on chromosome 16p. So that means that it's not only the quality of the DNA, but also the quantity of the DNA that matters. And that's a good example for chromosome 16, but there are also some other CNVs located elsewhere that probably also are also involved in the development of obesity. So the conclusion here is that you have different genes with point mutation associated or causing abnormality in the regulation of appetite, plus some others which do the same thing, but in addition have some consequence on the brain development, on the, and can be associated with psychiatric diseases and even with mental retardation. Now, what about common obesity? Common obesity is a vast majority of your obese patients in children, exactly the same in adults. And we know now that it's highly polygenic. Highly polygenic means that if you have a mutation in gene 2, for instance, the mutation is not enough to develop for the development of obesity. You need to have a different mutation plus exposure to the environment. How do we know that? We know that because of the Genome-Y Association study. We published the first GWAS study for diabetes and obesity 10 years ago now, and we have the protocol was a case control, or we analyze hundreds of thousands and now millions of polymorphism in large population where we had a quantitative phenotype, and one of the phenotype might be the BMI, and for children the Z-score of the BMI might be a good phenotype. That is a result of the latest result in a paper published in Nature by the big international consortium Giant. We are part of that as well as about 500 or 600 different other researchers. And we have collectively found more than 100 loci and genes with polymorphism that modify BMI and also increase the risk for obesity. There is also a consortium. I didn't show you this the slide. There is a consortium named EGG, specifically on children, but the number are smaller, but it's about the same gene that also increase the risk of obesity. I'm not totally sure, so that there's different gene increasing the risk for obesity in children compared to adults. So at least there is no strong evidence for common obesity. My point now is not about the number of genes, but what these genes are doing. So that is a figure of this paper based on bioinformatic analysis of the gene expression. So you can find in bioinformatics from public databases where the genes are expressed, and what this figure said is that different part of the brain, that most of these genes are highly expressed in the CNS and in the brain. Now, I'm always reluctant to show this slide because it's based on the very heterogeneous data. So we decided to do something, was to analyze the expression of the gene in a unique experiment in all the human tissues. Then you can find here, including beta cell, because we also look at the gene for diabetes. And for that, we use a technique named nanostring, which is quite interesting because you can count the number of copies of the RNA in a given tissue without PCR. So you don't have the bias of PCR, which is quite important when you want to compare one tissue with another. So doing that is the result that we had. And I think it's quite interesting. It's totally unpublished. First of all, the number of these genes that are expressed in the adipose tissue is very low. So there's not enrichment on obesity gene in the adipose tissue. You have some of them are in the beta cell, but most of them are in the brain. And as you can see, if you see carefully, it's not really hypothalamus. It's more the different region like insula, which is quite important for addiction, hippocampus, which is quite important for behavior, and substantia nigra, which is quite important for reward. So the conclusion here that the gene for monogenic obesity and polygenic obesity are not the same. For monogenic obesity is first appetite, second brain function, and for polygenic obesity is something related to our behavior, the hedonic component of food intake behavior, and what you do, what a child does when he's at school, when he has the birthday and the way this child behaves when he's stressed, and his relationship or her relationship with food. So we have to work more on that, but I think it was quite an interesting result to share with you today. This slide only to say that there are some other hepatitis that's quite important in child obesity, and we published this paper, and we have another one which in French population, that is the Mexican population, but which will be published soon, where we showed that the number of copies of the amylase gene is associated with obesity. And the children who have a larger activity of amylase were protected against obesity. And it's important that we found this effect in Mexico, because in Mexico, especially in the rural part, children eat 70% of their intake, calorie intake is in starch. And as you know, amylase is a key enzyme for the digestion of starch. What we found in adults, but we have to prove in children, is the way it increases the risk of obesity is probably through the microbiome. There is a difference in the microbiome of this of the individual with high amylase copy number compared to low, and that's maybe why there is this kind of protection of against obesity when you have a high number of that. So let me finish by what we should do. In my humble view, it's quite important to assess for monogenic or monofactorial obesity for every child with an hyperphagic obesity early in life, especially when there is some developmental delay, and especially if this child is from a consanguineous family. Now, how to do it? It's very easy these days. For instance, we published a paper in 2014 showing that with a strategy, a targeted strategy named Rendance, for about 200 euros, it is possible to easily test all the genes with a high quality, and that means to do a very good diagnosis of monogenic obesity. Now, the targeted thing is fine, but if it's something else, of course, you cannot do it. And what we have developed with others now is exome sequencing. And I think the exome sequencing, we can do that now, exome sequencing in my lab for about 500 euros. And it's good for diagnosis. We have extremely good quality, and also for research if it's another gene. Now, it's fine, but what about the syndrome? And what about chromosomal abnormalities? And you know how it works. You have a child with a possible syndrome. The first thing you do is to ask a geneticist to do a karyotype or FISH, or now a CGH array. And it's always a problem, because if it's not something, say, in the region of Prader-Willi, it's always difficult to establish causality. And it might be that there is a point mutation, and the CNV has no role. So we believe, personally, that what we have to do is to have a unique test. So we developed this test, and which it's based on very good capture, named MediWest from Nimlogen. Absolutely no shares in Roche, which work well for 99% of the gene, which is not too bad. In addition of that, we put on the capture kits CNVs, which means that we can detect abnormality of 50 kb or less all over the genome. And doing that, we verify that you can recognize with the same quality compared to CGH all the chromosomal abnormality. But in addition of that, we can look for mutation in the exon of all the gene. And what we do in addition now these days, yes, I finished. What we do now is the tissue-specific enhancer that can be useful. So today, what about common obesity? And to finish, I will say that there is no evidence yet that the genetic stratification is useful for the prevention or for the medical care of common obesity. But is it possible to predict common obesity? We published a couple of years this paper, which is absolutely not based on genetics, but on very simple clinical data and familial data. And as you can see here, we have a good specificity and sensitivity in the prediction of obesity in children in different populations. And what this slide shows that it's not sufficient to look for the BMI of the parents. You have 10% risk. You need to look for other factors that really increase the quality of the prediction, and it is really possible today to guess what is the target and who are the families we have to target to try to prevent obesity. And that was my last slide, and I want to thank the audience, the chairman, and the collaborators.